Welcome. Welcome, everybody. So glad to see a full house today. Welcome to this episode of the TIES webinar series. My name is Bertha Vasquez, and I am the director of the Teacher Institute for Evolutionary Science. And I am very excited to have our speaker here with us today. Her name is Sara Fitzsimmons. And I learned about this amazing story of the American chestnut tree by reading Overstory, which is a Pulitzer Prize winning book, and then reading an article about it in Sierra Club magazine. And it absolutely captivated me, this story about such an important American tree. And Sara was kind enough, when I reached out to her, to do this webinar for us. She has worked with the American Chestnut Foundation at Penn State University since 2003. Assisting chestnut growers and researchers throughout the Appalachian Mountains. Born and raised in southern West Virginia, Sara studied biology at Drew University in Madison, New Jersey. She received her master's in forest ecology and resource management from Duke University's Nicholas School. And after a short stint as an editorial assistant at All About Beer magazine, which I should have asked you about that as we were getting ready for this, Sara returned to the forestry field, where she has been ever since. She hopes her research and professional work will facilitate long-term conservation and restoration of native tree species at risk from exotic pests and diseases. We sure know about that here in South Florida. I will be monitoring the questions, which is down at the bottom where it says ask a question. Go ahead and ask a question, and I will ask Sara when she finishes her portion of this talk. I see quite a few students in here today. I want you all to pretend that Sara is a guest speaker in your classroom. Please keep the chat comments focused on the topic being discussed. Okay? And with that said, let me pass it on to you. I'm going to sit back and totally enjoy this webinar. Thank you for joining us.
Well, and thanks for having me and inviting me to talk about this program and what we're doing. I'm going to share my screen here. All right. Can you see that all right?
We can.
Okay. So I'm going to cover the basic story about the American chestnut, talk about how we got to this point of why I'm hired to do this work, then what we're doing to try and restore the American chestnut back to its native range, and how we're doing that. The American chestnut was a very important species, particularly in the 1800s. It was a major component of the eastern forests. It grew really fast, large, it's highly rot resistant, a highly versatile timber species. And what I've heard it called is a cradle to grave species. And that means that you could use it for cribs, coffins, and everything in between. And, the American chestnut didn't do any one of these things the best. It's not the fastest growing tree in the East. It's not the biggest tree in the East. It doesn't produce the most food for wildlife of any species, but it does all of those things really well. And I can't think of another species that has this whole package. And the nuts were incredibly valuable to wildlife. And you can see this timber form here on this picture right here. You have a person standing here at the bottom of this tree, and there's not a branch up until 60 foot. That's, pure timber type. These things can grow up to an inch or even 2 inches on the best site, 2 inches in diameter a year. So really, really fast growth, even if it isn't the fastest. On the right, you can see the native range of the American chestnut, and this was taken from a U.S. Forest Service publication from the 70s. So it's not perfect. Don't take these lines as definitive, but it's from Maine to Georgia, west into Indiana. And you can see, little bits and pieces in the Piedmont of Virginia, North Carolina, down into Georgia. So it certainly had quite a big range, and that covers about 180 million acres. Lots of historical uses. Again, this real jack of all trades kind of a species. Wood products, agricultural uses with the nuts. I love this picture in the lower right-hand corner. If you can read the engraving on the roaster, it says, "Hot chestnuts, good for the brain, try a bag." And I think, people are always looking for things that can give you good health benefits. It is true with chestnut. Up above that picture, you see chestnut leaves, and there are some antiseptic qualities of the tannins that are in chestnut leaves and bark and things like that. But I think most people are more familiar with the wood. And so you can see a split rail fence here. I think this is from Virginia, from southwestern Virginia. There's lots of social and historical connections. Chestnuts roasting on an open fire, of course, is one of the one that most people know about. And you know, it's so closely tied to society because of its utility, because it can be used for so many of these things. And there's some global connections too, which I'll get to in a minute. This is what the species used to look like. So this is a picture from Shenandoah National Park in 1912. I don't know if you can tell, but right across the middle slope, at the bottom of the slope up toward the middle, you can see these white treetops. Those are the male flowers of the American chestnut. They look like long, fuzzy pipe cleaners. They're called catkins. And so some people would say that it looked like it was snowing in July. That's when the male catkins are at their height. And so you can think when 25% or more of your forests were chestnut, there were an estimated 4 billion American chestnut trees in the 1800s. And you can see very densely populated. You can see how one could say, yeah, it does look like snow in July because of all of these flowers in the treetops. So what happened? In the 1800s, 1700s, 1800s, there was a lot of trade of plant material. People were bringing in lots of stuff from Europe, lots of stuff from Asia, because they were exotic, beautiful plants. I mean, we see them all over the place today. A lot of that was happening in Philadelphia, New York, New Jersey. There's a lot of really phenomenal gardens that had been established, Longwood Gardens, Mount Cuba Center, you know, a lot of the DuPont legacy in Delaware and the Philadelphia area. But what people didn't realize is when they were bringing over this stuff, they were also bringing over pests and pathogens that the species here had not seen before. And in the case of chestnut, the American chestnut, there was a fungus that was being brought over, now called Cryphonectria parasitica. And it was brought over on Japanese chestnuts, actually. So Japanese chestnuts had been imported for decades into the U.S. If you look at the New York Times or papers out of the Philadelphia area, you'll see nurseries advertising, you know, beautiful Japanese chestnuts, and they really are striking. If you see an old specimen, they're a beautiful tree. But that's what brought over this fungus. And Japanese chestnuts, the Asiatic species have resistance, and the American chestnut does not. And so what happened is you have this disease that spread across its range very rapidly. Even though it was in here in the late 1800s, it wasn't identified until 1904. And so that's what you see on the map over here on the right. You see 1904, New York City. That's when a zoologist at the Bronx Zoo isolated the fungus, put it on a petri dish, performed Koch's postulate, figured out, okay, this is what's killing the chestnut trees here in the Bronx Zoo. He called it the chestnut blight, the chestnut bark disease. And what happens is this fungus spores get into an entrance in the bark. It could be anything. It could be a wound. It could actually just be the bark maturing. You can see here in the picture with the orange.
Just a sec. We lost your screen share just a second ago.
Okay. Let me get back in.
Just try to do that again.
Okay. Sorry about that.
No problem.
Are we back?
No, that's so weird.
No? All right.
There it is.
How about now?
Yeah, you got
it. All right. So the fungus gets into the cambium layer, the living layer of the tree, through any sort of type of wound. You can see in this picture with the orange canker, it's coming right at where a branch comes out. That's enough for the fungus to get in. It's just that simple, what you call a crotch in the branch for the spore to get in, germinate, and create this canker. What the good thing, I guess, if there's a good thing about chestnut blight, is that it doesn't kill the roots. It only kills the above ground portion. That's a whole other presentation about why it doesn't kill the roots, and we can talk more about that if y'all are interested. But it's important to know that it doesn't, because that's the only way that the species has persevered, is because it only kills the above ground portion. The roots remain, and it's able to resprout. And here's a picture of that. So you can see all the orange sporulation here You can see the cracking of the bark in the bottom. The fungus eats the living layer, the cambium layer. So what the fungus does is and this is important later, the fungus exudes oxalic acid. The oxalic acid kills the chestnut cells, and then the fungus eats it. This fungus is a necrotroph, so it likes dead things. So it can even stay, once the tree dies, the fungus is still sporulating and very happy because the tree's dead. It doesn't have to kill it. It can just feed on it already. So all this sporulation is just the fungus creating more spores and spewing out into other chestnut trees. And what the chestnut tree does is it dies back, the roots send up more sprouts, and it dies back and it sends up more sprouts. And you have these sprout clumps that can persist indefinitely as long as it has, food and light and doesn't get eaten by deer. I'm just showing where all the spores are here on that tree. Now, this is what it looks like when you have, a very densely populated chestnut forest die of chestnut blight. These are what we call ghosts of American chestnut. This is a picture from the mid-1900s in Shenandoah National Park, where there were millions of American chestnuts. And you can see how devastating that would have been, especially for a species that was so, that in an area where people were so dependent on a species that served so many purposes and such great utility. Today, this is pretty much what you find in the eastern US. So if you hike the Appalachian Trail, if you go into mountains, particularly in Pennsylvania, North Carolina, Massachusetts, even, you will see this all over the place. You see, the dead stems in the middle with sprouts coming up around it, and then the very classic chestnut leaves close to the base. A common misconception about the American chestnut is that it's threatened, endangered, or extinct, and it's none of those. And the reason for that is that there's an estimated 430 million trees still in the landscape. That number was come about because of an evaluation and extrapolation from FIA data. FIA is Forest Inventory Analysis. And the map for that is here on the right. This was done by John Scrivani. He's a great geographer and forester in Virginia. But you can see tons and tons of sprouts, especially the national forests of western North Carolina and eastern Tennessee, the Shenandoah National Park. Pennsylvania is just covered in them. And then even, somewhat surprisingly, Connecticut, Massachusetts, New Hampshire, there are sprouts all over the place. Because of the sheer number of these sprouts, it's not threatened, it's not endangered, it's not extinct, because there's 430 million of them. But 84% of them are less than an inch in diameter. So they all look like this little guy here in the lower left-hand corner. They're not performing their function, either economically or ecologically or culturally. They're just this sort of, they just exist. So that's why we call them functionally extinct, because they're no longer performing the function that they need to in the forest. Well, previous to their functional extinction, it was a native species that was important to the greatest food web. And I think that, there's another scientist, Doug Tallamy. He's an entomologist at Delaware, and he's really brought the importance of native species to light to a lot of people, and really made how important native species are by observing insect herbivory on native species, and how you can see a 100-fold increase of insect species on native plants versus non-native. And there's a reason for that. People like non-native species because they don't get eaten by our native insect species. And so a lot of people plant those because they don't have to care for them a good deal. But the opposite side of that coin is you're not feeding our native wildlife and fauna. So I love this picture because it shows all the different kinds of creatures that actually feed on chestnut, or on something that feeds on chestnut. How about fish? How do we get there? Well, that's because the leaf litter decomposes and allows freshwater larvae to feed on that decomposing leaf litter, and then a fish eats that larvae. So that's important. You've got bees that feed on pollen. In fact, there was a thought to be extinct chestnut bee that was recently found, because chestnuts have become part of this work to try and restore the species, and has been found again. And we're doing some volunteer work to try and find the chestnut bee and monitoring and see where we can't find it in our research plots. And I think the others are pretty self-explanatory. Bears, grouse, chipmunks, they all eat the chestnut itself. And just to give you a sense of dependable mass production, chestnut typically produces chestnuts every year. This is opposed to oaks, also in the same family, in the Fagaceae. Oaks tend to be more cyclical in their production. So you tend to have, maybe every 3 years you get a really heavy mass year, and then you get reduced mass set years in between, whereas American chestnuts typically produce pretty high mass crops every year, and that's important.
Strange. Sorry, again, we lost your screen.
Oh, geez. I'm sorry about that.
I told you there's never a webinar without a tech issue.
Okay.
It has to happen.
Yeah. Well, thank you everybody for bearing with these issues. There was a study that was done in Western North Carolina, which suggests that when chestnut was lost from the forest, we lost 34% of mass production for wildlife. And so while nothing necessarily went extinct because the chestnut was extirpated from its function, we certainly lost a lot of resources for not only wildlife, but also people. So how did we get here? What is it about American chestnut? Why doesn't it have resistance when other species do? How does the Japanese chestnut take the chestnut blight and bring it over here? How can it survive with this fungus? So let's talk about chestnut as a genus. This picture doesn't show all of the different species in the genera, but depending on who you talk to, if you talk to the lumpers or the splitters, you can have at least 7, but up to 12 species in the genus Castanea. Most commonly, you're going to find the 5 here. The American chestnut and the Allegheny chinkapin are native to the eastern U.S. The Chinese chestnut you're going to find all over the place because that was imported as a way to restore the American chestnut, actually, in the early 1900s. European chestnuts, obviously native to Europe, but were brought over to the U.S. and planted pretty extensively. And then the Japanese chestnut, of course, which I already mentioned, is what actually brought the blight over. So you can find these, especially the American and the Chinese, you're going to find them a lot. And you can see there's a pretty big difference in their leaf shape. And there's a lot of other qualities too we won't get into. Again, that's another hour workshop of talking about chestnut leaf ID and morphology. But what I think is really interesting is what is it about these genera that can, how did the American chestnut get here? What's the center of origin of this genus? And not surprisingly, the center of origin for the genus Castanea and also the Cryphonectria parasitica is China. And there's a lot of similarity between temperate Chinese forests and temperate U.S. forests. And that's why actually we're having so many problems with invasive pests and pathogens. I was able to go to China a couple of times, and what fascinates me is how similar their forests are. I mean, you would not believe that you were that you were not in the Southern Appalachian Mountains. I mean, you are able to identify, if you know the species in the Southern Appalachian Mountains, you can identify virtually every species, at least its corollary, in these central Chinese forests. So these pictures are all taken from an area called Shenandoah, which is in central China. This is the Chinese basswood, the tulip poplar, their sassafras, a Hercules club there on the right, something that looks very much like a chestnut oak, cherry, tulip poplar, virtually indistinguishable from their American counterparts. I mean, unless you get into really nitty-gritty morphological characteristics. And I think that's fascinating. Why are our forests so similar, and how did these species go from that center of origin to get over to the U.S.? I think a lot of people, the first thing that comes to mind is the Bering Land Bridge. Well, it must have come over from China up through Russia, across the Bering Land Bridge into Alaska and down through the western U.S. And that's possible. We have not ruled that out with chestnut, but the preponderance of evidence, particularly molecular evidence, suggests that there's actually a westward expansion of the genus, that the center of origins in China. You had a split of the genus there long before it went west, but you have 3 different species in China. You've got an early split into Japan, and then later splits going to Europe, and then using the North American land bridge going across the Scandinavian areas and getting into the US, and then subsequent speciation after that. So just to give you in a sense, the suspicion is that this happened in the between the Eocene and Miocene time periods. And so you can see here, here's Europe. You've got so the potential land bridge here going across Greenland and in North America. Here's the Bering land bridge. Again, we haven't ruled that out. That's certainly possible, but most likely it's a westward expansion. And another reason we think that too, you got to look at diversity of the species. And so this is one of the really, really early studies that was done. I'm going to hide that. That was done on diversity of the Castanea genus. This was done in 1994. It's an allozyme analysis. And you can see down here just overall average number of alleles per locus, or even the estimated heterozygosity. You can see here's Millissima, the Chinese chestnut. You've got average number of 2 alleles per locus. That's reduced in dentata, and then your overall heterozygosity is almost half of that in American chestnut as it is in Chinese chestnut. So this is expected. As you've got a split from a center of origin, you're going to have more and more reduced diversity. And so let's kind of poke on this a little bit. Like, what is it about within the American chestnut genus or species itself? How much diversity is there actually, and where are the diverse points? And so in 1998, there was a follow-up on that study looking at the different species, and you can see here sort of some population structure showing up. So here in the center, you've got a lot of
Sorry, screen share again.
Dang. All right.
I don't know. That's never happened before, but I think every teacher watching knows what it's like these days. So
yeah, very frustrating. Sorry. Well, so in the center here, you see Ohio, Michigan, North Carolina, Virginia. You've got this sort of central, not a lot of diversity here across the area. You've got a northern sort of population, New York and Connecticut. You've got some Pennsylvanias maybe breaking off. But what's really interesting is this Alabama population really breaking off. And this is important. And you'll see here in the next study that was done in 2006, looking at a lot of additional locations, getting up into Maine, looking at a Canadian population, looking at Kentucky. Based on that information, and this chart actually doesn't show you much, this was built into eventually discovering that a majority of your diversity is down in the south. So the red indicates much more diversity. The green is a single haplotype, what we call D germplasm, quote unquote. And this isn't surprising that you find most of the diversity of the American chestnut in the south. And the reason for that is because during the last ice age, most eastern U.S. tree species sought refuge from the glaciers in the southern Appalachians. And so as those glaciers retreated, you have this very small-ish founding population in the southern Appalachians. You have maybe 10,000 years for these things to start marching northward. In the case of the American chestnut, it didn't go as fast as other species. You have a large nut that doesn't go as fast as some other type of seeds, and oaks were able to disperse much more quickly and even find other refugia. So chestnut has not had enough time to give the northern populations. They haven't been able to break out of that genetic bottleneck, and you've got much less diversity up there. You've got some regional adaptation for cold, but that's about it. By and large, a majority of the diversity of this species is in the Southern Appalachians, Tennessee, Mississippi, Alabama, Georgia. You find a huge amount of diversity down there. And so kind of, let's put all this into perspective. It's great to know about the diversity of the species. Why do we care? Well, mainly because that's going to be a driving factor for how we restore the species. We've got a species that served all of these functions that has become all but functionally extinct in the landscape. How do we bring it back? So you've got one aspect of where you need to get disease resistance back in. You need to give it the tools to face off this fungus and be able to resist it. Then you need to get it back in the woods. And so while those two goals are intertwined, they also rely on different types of information. So let's talk about disease resistance. How do we get that into the tree? How do we give the tree the tools to fend off this fungal invader? And what the American Chestnut Foundation does is works with what's called 3-burr. And I like that. There are 3 nuts in a chestnut burr, and the burr is what contains the seeds. And the 3 Bs are breeding, biotechnology, and biocontrol. And those 3 Bs are united for restoration. So 3BUR. And you can see here in the lower left-hand corner, that's a traditional breeding. These are controlled pollination bags. The picture in the middle is for biocontrol, and the one on the right is for biotech. Let's talk about breeding real quick. Breeding is what the American Chestnut Foundation started in 1987, and they actually built on breeding programs that were started by the USDA back in the 1920s. So they took material that was the best of that program and then bred additional generations to try and find the right combination of blight resistance and American character. This is the seed orchard that I work on here at Penn State. This is in the Arboretum at Penn State. And you can see all the huge number of stems on the left, very few on the right. The ones on the left, we only select less than 1% of what we plant for blight resistance. It really is looking for, it's very complicated. Blight resistance is very complicated, more complicated than we expected. And even still, we were only selecting less than 1%. So all those little pink dots at the base of a tree are a tree that don't have resistance. So we cut them down, we throw them on the bonfire. Trees that live, we let them grow, and we examine how their blight resistance holds up over time. The trees on the right, we actually take the fungus, we put it in the tree. It's a process called inoculation, and you can see there are cankers at the base, but the trees are resisting it. They're pushing that fungus out. The trees on the right have been living for about 20 years now with this infection. So it's not perfect. It's not quite Chinese level of resistance, but it's certainly better than American chestnut levels of resistance. And it's a piece of the puzzle of getting these things back in. And as far as American character, they look pretty good. So if you look at the form and architecture of these trees, they are growing quickly. They grow with acute branch angles. They're growing tall and straight, and that's what we want in terms of American form. Another B, the biotech piece. So I think whenever someone says biotechnology, they immediately think about genetic engineering, and that is a piece, and that's what I'm going to focus on. But I also want you to know that biotechnology encompasses other factors. So there's marker-assisted selection, there's genetic sequencing, there's all these things that can inform other aspects of a restoration program. CRISPR, gene editing, is certainly something that we're looking at. But the most promising avenue right now is the insertion of the OXO gene. And so y'all remember when I was talking about what the fungus does to eat the chestnut? The fungus exudes oxalic acid to kill the chestnut cells to eat them. Well, this OXO gene produces oxalic oxidase, which neutralizes oxalic acid. And it's interesting because OXO can be found in many, many different plant species. This particular OXO gene comes from wheat. And so scientists at SUNY ESF discovered this, put 2 and 2 together. It's primarily Bill Powell at SUNY ESF. He also worked with Chuck Maynard, who's since retired. But they discovered that this OXO gene should be the key to giving blight resistance through biotech and genetic engineering. how do you get the gene in there? They looked at many different techniques, gene guns, various different things, eventually settling on something called agrobacterium-mediated transformation. This is a fascinating technique. I am not joking when I say you should look it up because it's really fascinating. Agrobacterium-mediated transformation uses Agrobacterium tumefaciens. And the reason I like to put this picture on the left, Agrobacterium tumefaciens.
Share your screen. Okay. I think, Matthew P., one of our attendees, is noticing it happens about every 5 minutes. So I think your screen might be falling asleep. So just move the mouse around every once in a while. I am
actually.
You are.
Around, I think. But,
there's like a timer or something going on.
Okay. I'll be a little bit more. I tend to talk with my hands.
I'm only stopping you when you say because the picture on the left or whatever. That's when I realize
Okay. So the picture on the, so you can see it now, right?
Yes, we can see it now.
Yes. Okay. All right. So Agrobacterium tumefaciens is a bacteria. It's the causal agent of crown gall. And again, the science of that is fascinating, but just take my word for it that that's how you get this gene into American chestnuts. You take chestnut embryos, you put them in a petri dish, basically with agrobacterium and the plasmid that you put together. You put them in tissue culture, and they look like little palm trees in these tissue culture boxes. You wait 18 months, and then you put them out in the field. This is looking very promising. It does give certainly elevated resistance. It looks like it's close to Chinese chestnut levels of resistance. That still needs to be tested in the field. The difficulty with biotech, and many of you are probably familiar with this, is that anything that's genetically engineered is very tightly controlled through governmental regulation. These specimens, materials are regulated by 3 different agencies: the USDA, the EPA, and the FDA. And in order for them to be planted currently, they need to be under permit by the USDA. And there's a lot of controls on that. They can't be allowed to flower. If you do, you have to put these screens over, you see here. They can't be allowed to grow over 30 feet in height. You have to submit monthly and annual reports. You have to get a new permit every year, although we might be able to get away with 3 years now with the new system. None of these are going to work for restoration. In order for them to be, and you can't plant, the EPA regulation is that you can't plant over 10 acres of material. So again, if you want to restore 180 million acres, you can't do that under any of these very tight regulations. So what you got to do is get them deregulated. And that's the next step. So TACF worked very closely with the scientists at SUNY ESF to get the petition up on the Federal Register to petition the USDA for deregulation. We're currently working to get the EPA petition in to get it deregulated, and then the FDA will come shortly thereafter. And there's a public comment period on all of these. The public comment period for the USDA was in the fall. 62% of the comments were positive for deregulation. It's not a popularity contest. I only say that because I think it's pretty cool that lots of people, we had over 4,600 comments come in, and that's really high actually for a genetically engineered material. But what these regulatory agencies are looking for is what's called a substantive comment. So you can say, I love chestnut trees, and yes, that's interesting, but that doesn't go into their decision. You can say GMO trees are going to destroy the ecology of the world, and that's not going to go into their decision. They need what they call substantive information that gives them scientific basis for them to say, yes, it's deregulated, or no, it's not. There's going to be another couple public comment periods that come up probably within the next year. The USDA will have another 2. The EPA will have 1. We'll certainly have 1 from the FDA. And it looks like we might have an answer by mid or end of 2022 is kind of the timeline we're looking, but we have no idea. It's really difficult to make any sort of definitive judgments in timeline, but we're hoping that by 2022 we'll get an answer, and we're optimistic that it will be favorable and that it will be deregulated. whenever I talk about genetic engineering, the first question that comes to mind that people ask is, what about the safety? And you can't say without a doubt, you can't possibly research every permutation. That said, everything that we've looked at with ESF and other partners, there's no difference between a transgenic American chestnut and a non-transgenic American chestnut in terms of their function. Leaf litter decomposes the same and feeds wildlife the same. The germination of seedlings does the same. The critters feed on the leaves the same. Mycorrhizal fungi, that was a big concern because you've got an antifungal agent, the oxogene. Will that decrease the amount of mycorrhizal fungi colonization? It does not. You have similar between transgenic and non-transgenic. Bees feed it on the same. There's no detrimental effect on bees and their feeding on transgenic versus non-transgenic pollen. Tadpole development, they develop exactly the same after feeding on leaves in transgenic American chestnut versus non-transgenic. So all of these studies speak toward the safety of this technology in American chestnut. The next step, and which is what we're working on right now, is diversifying transgenic. So the transgenic tree, what's asking to be deregulated, we call it Darling 58. Darling 58 is a single event, it's a clone. And restoration, you don't just want to submit a clone. And that's why I talked a lot about diversity and genetic diversity and in the American chestnut. You can't just submit a clone and have it take over for a lot of reasons. And I'm sure you guys have talked about that in other aspects of animals and plants. Oh, see, I was moving the mouse. So
I believe you. It's just I'm going to interrupt you every 5 minutes and say screen share.
That's fine. Dang. If anybody has any other ideas, though, throw them out there. So we want to diversify Darling 58, and we do that by using the remnant populations of American chestnut. We document their locations. If y'all want to go out and find some trees, download TreeSnap, go out and tell us where you found an American chestnut. We then collect nuts from them if we can, or graft them, or transplant them, or air layer them. We just try to get them into what we call a GCO, or germplasm conservation orchard. And our goal is to diversify Darling 58 on a minimum of 1,000 wild type Americans for then spreading it out into the native range. So those are two B's. The third B is biocontrol. And again, there are other things that we can look at in terms of controlling the fungus. None of them are incredibly successful, but we're investigating them nonetheless. The most promising avenue looks to be what's called hypovirulence. And hypovirulence is what happens when the fungus gets sick, it gets infected, it itself gets infected by a virus. And so the bark on the left is an uninfected tree. That's a tree that has not been infected. It's not resistant. It just is lucky it hasn't become infected. The one on the right, you remember those pictures earlier where I showed like the real sunken, orange, sporulating, ugly cankers? Well, this, depending on your perceptions, might actually be uglier, but it's a non-lethal canker. What happens is the tree is able to put down basically what it's equivalent to a scab, and it pushes the fungus out, and it's able to survive. It's big, it's ugly, it's scabby, but the tree lives. And so, and then here on the right, you can see these petri dishes filled with chestnut blight. The deep orange are virulent, non-infected strains of the fungus. The ones with that white top, these are strains that are infected with the virus and the hypovirulent Cryphonectria. And so this is another tool. And what I've been working on this for a long time, and in talking with all of my colleagues, and I love this 3-burr approach. And the 3-burr also doesn't necessarily negate anything else that's out there. But as with any of these large scale problems that take over lots of time and lots of space, none of these solutions, not even biotech, not even OXO, is going to be the silver bullet of what restores this species. It really is going to be a combination of all these techniques. in addition to what's coming afterward. So we're looking at CRISPR, we're looking at other events, we're looking at other genes, because as great as OXO is, we haven't put it to the test in the field because of regulatory issues, and you still want a more, you've got 180 million acres that you need to restore. We're going to have time to look at additional tools to give the species lots and lots of tools that it can go to over the millennia. We hope that it will be in the eastern forests. The way we do that, there's a broad, what I love about the American Chestnut Foundation and all the people that work with it is that it's a broad network of partners, scientists, and volunteers. TACF was formed in 1983. There's over 5,000 members and 16 chapters, but we can't do it alone. We mainly just try to help our partners do the work that will restore this species. We work internationally with universities in China. Obviously, ESF is a huge partner. I'm at Penn State, and we have a great working relationship with that university and many others. The Forest Service, the National Park Service, all of our state forestry agencies have been phenomenal partners. TACF cannot do it alone, and it really is through, and I'm not even touching on the citizen scientists that do this work. Jim Hissam is there in the lower right-hand corner with one of my past interns, Shuzhen Yu, and we have over 500 orchards, at least half of which are on private landowner land. And when you look at the breakdown of land ownership in the eastern U.S. in the native range of the American chestnut, 80% of that is owned by private landowners. And, we work really well with the Forest Service and other public land agencies, but restoration is going to be driven in a huge part by private landowners, people who have 10 acres of woodlot or 1,000 acres of woodlot and help us get the species back on their land. There's a lot of additional challenges that, again, that's yet another talk that we can get into about the challenges of actually getting the species back into the woods. There's other diseases out there. There's invasive competitors that are going to be a huge roadblock for us. There's 180 million acres. That's really hard to conceptualize, how we're going to get the species back across that entire native range. And then if you talk about climate change, how is that going to impact our restoration plans? But maybe we can talk about that another time. Again, that's a whole other presentation. And I kind of want to get back to this idea of the partnerships. I'm really excited. There's a lot of talk about genetic engineering and its impact, and a lot of people like to say how indigenous peoples will be poorly impacted by the application of genetic engineering, and I'm really excited by our recent MOU signing with the Eastern Band of the Cherokee Indians. Again, these are people whose lands are right in the heart of the range of the American chestnut. And they've recently embraced this technology and are looking forward to the day when transgenic trees, blight-resistant trees of any kind and any background can be put on their lands so that that species can again be used for cultural heritage purposes. I don't know if you guys are familiar with Cherokee chestnut bread, but chestnuts can be ground into a flour, which you can then make gluten-free products, gluten-free beer, gluten-free bread, all of these things. So there's a lot of really amazing uses, both current but also historical. Again, it's really going to come down to these amazing partnerships with and people driving this work. It can't be just one agency or one organization alone. It's going to have to be hundreds and thousands of people working to get a species restored. We record over 25,000 volunteer hours per year. Do I have to share again?
Screen share, yeah.
All right. I see. I almost predicted it.
I was looking for a good spot to interrupt you.
Yeah. All right. So we record over 25,000 volunteer hours per year, and this is doing any number of things from finding trees to planting trees. The genetic engineering, you can't really do that in your kitchen just yet, but virtually every other part of the project, we work with amazing people to get different parts of the job done. I love this quote. This is Gifford Pinchot. I wish I could come back in 100 years and see my trees. And that's why the people that me, in fact, I don't think I'm going to see the product of this work. But I know even the people who started this work with the U.S. Forest Service and the USDA in the 50s, you know, you work on a tree breeding project like this, knowing that you may not see the results of it because it takes so long. It is such a large scale temporal and geographical problem that you're doing it for your kids, grandkids, and for, you know, the greater good. With that, I will end there. I love this picture of a chipmunk with a chestnut, and I'm happy to take any questions that you all might have.
Wow. I'm not going to do it, but I could take the next 10 minutes and just ask my questions. First of all,
Okay.
If you come to South Florida, you must call me. I will take you to the Everglades, completely different habitat ecosystem, but I would love to spend more time with you. I loved listening to you. Let me ask you some. First of all, those of you asking if you have access to the presentation, this will be recorded. It automatically becomes a recording when I stop the webinar, so that's fine. So what are the chances a person hiking in the eastern U.S. comes across a chestnut tree?
It depends on where you are. So, you know, I'll go back to that map that John Scrivani made with the FIA data. If you're on the Appalachian Trail, or, you know, on a, you know, in short sight of it, like here in central Pennsylvania, you hike Mount Nittany, you go on any game lands, you go on the Appalachian Trail, you're going to find hundreds, if not thousands, of trees. In other parts of the range, in southern Ohio, which is part of the native range, and where I've really been trying to find trees in southern Indiana, where we've got amazing reports of huge trees in southern Indiana, you can't find them anymore. And that's one of a big northern Mississippi, huge part of diversity. We really need to find trees in these locations, but it's very difficult to find them. So that's a mystery. Like, we think we know why we're not finding them. Probably a land use issue. But we haven't proven that. But in those areas, it's very difficult to find them, but those are the most important locations to find them.
Have forestry services, both state and federal, looked into planting federal and state land with chestnut trees?
Yes, and we have planted chestnut trees with municipal all the way up to federal public lands. The transgenic is not yet anything because of the permitting and regulation, and there's other certification issues that go with transgenics, but we're hoping that when deregulation comes along, we can attack other, well, I use that term loosely, we can work on other certification challenges to get them in the future.
You covered this a little bit, but is there a fungicide that can combat the spread of the fungus?
Not really. There might be a new one that's on the market that we're actually trialing this summer. It's not labeled for it, so I'm not going to say it, because even if I say it, you can't use it on chestnuts because it's not labeled for chestnuts. But everything that's been tried to date doesn't work, or if it does, it works for a year or two and then stops working. So no, unfortunately, there's no fungicide for chestnut blight.
Here's somebody from Oregon. Makes sense she's asking this question. If it came across the Bering, seems like there would be more similar species in the Northwest West Coast.
That's a really good question. And so there could have been species that have since gone extinct. And, you know, the so-called missing link that we don't know about, that are gone from the record, because it hasn't been long enough to have fossils of those species. There is a closely related genus called Castanopsis. Castanopsis is on the West Coast. The golden chinkapin is out there. We have not really evaluated that and how closely related it is. So it's the North American land bridge is the most likely scenario, but we haven't completely ruled out the Bering land bridge either.
Okay. I know it's a pretty fast growing tree, but this question is, have you considered using genes that speed up the chestnut growth? I guess at this point you're more worried about keeping it alive.
Yes. And who knows, maybe in the future, once we get the diseases taken care of, we'll look at growth factors. But at this point, that's not a major concern.
I don't know if this is a tree species. It's not something I'm familiar with. Does this blight infect privet? Is that a tree?
No, it's a woody herbaceous invasive.
Oh, okay.
Plant. Yeah, it does not, unfortunately. Cryphonectria parasitica is pretty species specific. It can infect some oak species, but it's rare for it to infect oaks. It doesn't infect any other genera or species to my knowledge. There's other species of Cryphonectria that affect, like there's a Cryphonectria cubensis affects eucalyptus, and there's a couple of other Cryphonectria species, but they're pretty host specific.
Why is there so much diversity in Canada? Is there?
There isn't. It also could be, and that might be an anomaly of stuff that's planted up there, stuff that was taken from the south and planted up there. But for the most part, there isn't a lot of diversity in Canada per se.
Do you have one planted in your yard?
On my 0.3 acres, unfortunately, no. I live on a woodlot, and I don't know if you can see my rhododendron, but I have a couple white oaks and a couple red oaks, and that's about all I got.
We got a couple people saying, well, how do we comment? What type of comments for the public commentary period? What type of comments can we put in there for you, or do they have to be more scientifically specific?
Well, I would say any positive comment doesn't hurt. And I would encourage you to just go on and say, I'm supportive of this. This is great. And that's mainly just from the standpoint of knowing that, yes, there is support out here for that.
Where, do you have that website, or can I type it into the chat for people?
So if you look up Federal Register and deregulation and American Chestnut, you'll find it. You'll either find the TACF site that gets you there, or you'll find the Federal Registry. All comments on all deregulated species are public, which is it's fascinating to go through and look at all the corn species, bioengineered corn species that are out there, and read people's comments, both positive and negative. As far as what constitutes a substantive comment, something that helps the USDA or the EPA, they're looking for data. They're looking for direct observations that will help them make a decision. So, for example, one of our members was the first consulting forester in Indiana, and he has firsthand accounts of putting chestnut in the landscape for 30 years. And so he can speak to how chestnut is not at this time invasive, and that's a concern. Will a transgenic American chestnut become a plant pest risk? And so they want data. It doesn't necessarily have to be statistically analyzed, but they want data or case studies or information that showcase that chestnut will not become a plant pest risk and or is not a danger to other species that are endangered. So that's the kind of information that they're after.
Okay. More questions are coming in, but I do like to keep these under an hour. I think that's a bite-sized amount that people can handle. I have one more question. I don't mean to shift back to the West Coast, but curious if there's any comparison studies to sudden oak death. That sounds terrible, by the way. Sudden oak death affecting oaks through California and Oregon.
Sudden oak death is awful. Sudden oak death is caused by an oomycete called Phytophthora ramorum. And
boy, you should go on Jeopardy. You are like...
If it's about chestnut, hopefully I know about it. But Phytophthora ramorum is related to Phytophthora infestans, which is the Irish potato blight. Also related to Phytophthora cinnamomi, which killed chestnuts in the Piedmont of the South. It's a whole other critter from Cryphonectria. It's not even a fungus. It's an oomycete. It's a water mold. So not a whole lot of parallels there other than please don't bring it here.
Yeah, we don't need anything else over here on the East Coast. Sarah, keep it over there, Sarah. Sudden oak death. That's my new password for my Bank of America account. Sudden one oak death smiley face. Wow, that was great. I have to get on a plane to volunteer for you guys, though, because, not happening in South Florida. But I really, I was looking forward to this webinar. It was everything I hoped it would be. We may, Sarah, have to get you to do some of these presentations when I do ties workshops in places like North Carolina, where it's very, very relevant, and the teachers can take the kids outside and talk about the history and the coevolution of the Japanese or Chinese chestnuts and the artificial selection. I mean, there's a lot of evolutionary topics involved, and it's so, so relevant. So I want to thank you so much for joining us and taking your time. Yes, we have a science leader from Winston-Salem, Forsyth County, who is like, yes, North Carolina. Another reason I'm coming up there, Iris. We're going to do this up there. And we could go look for chestnuts together.
The American Chestnut Foundation's main offices, administrative offices, are in Asheville, North Carolina. There is tons of chestnut out there. Lots of really great resources, both with the Forest Service and other aspects. But yeah, that's a great place to do chestnut work and learn more about it, and also get people involved and excited about this work.
Excellent. Iris, I think I'm going to have to do a full day workshop up there. Just saying. All right. Thank you. Thank everybody for joining us. This will become a recording once I turn this thing off, and everybody have a good evening. And thanks again.
Thanks, everybody. Take care.